Hello. So this is where we left off with the last video. The basic enzymes we've discussed so far in reference to DNA replication. That's making a new molecule of DNA before we go into cell division. So we're in the S phase of the cell cycle, synthesis between G1 and G2. So we have to copy our DNA in order for it to divide. And the first step is that helicase enzyme. Notice anything that ends with ACE is going to be an enzyme. Helicase, on the other hand, is an enzyme that's going to break apart this double helix right here. So you can think it starts with HELI, just like the term helix. So helicase enzyme uncoils and unzips these hydrogen bonds, causing DNA to split apart within that nucleus. The next enzyme is DNA polymerase. Again, ACE, so you know it's an enzyme. It's an enzyme that makes a polymer. This polymer is DNA. The polymer of DNA is made of smaller repeating subunits known as nucleotides. So it moves along each individual strand and connects complementary bases. So our base complements thus far we learned about were A and T, that's adenine that bonds with thymine, and then cytosine and guanine, or C and G, so A, T, G, C. Now, the final product being two identical copies of the original DNA molecule. Now, it'd be really simple if it worked the same way in each strand, but that's not necessarily the case, which is what we're going to talk about today. So, the Messel's and Stahl experiment showed that DNA replication is semi-conservative, meaning every strand of DNA is half old, the blue, and half new, that's the yellow. You can see the same thing over here, only with blue and pink, the blue being old, the yellow being the new. So every time it goes through a replication, they found that there is the same amount of original DNA, but new DNA increases and increases and increases. They use this using radioactive nitrogen to build the nucleotides, and they acted as markers so they could track them throughout different levels of replication. Every time that cell divided, it first replicated its DNA. So Messel's and Stahl is something we'll talk about at a future date. Let's talk about the replication fork. So replication fork is this part right here, the part where helicase actually unzips that DNA molecule. So DNA runs anti-parallel. Parallel meaning it runs side to side like this. Anti-parallel means it runs side to side, but in opposite directions. We label these directions 3 and 5. That comes from our organic chemistry background, where we actually number the carbons in a clockwise fashion. Carbon 1, carbon 2, 3, 4, and 5. This is a simplified model of deoxyribose sugar, a 5-carbon sugar, 1, 2, 3, 4, 5. And you'll actually notice that these are there, 4 is there, but it bonds in a 3 to 5 direction on one side. The other side, it's actually inverted or flipped, and it bonds in a 5 to 3 direction. The 3 to 5 is called the leading strand, whereas the 5 to 3 is a lagging strand. DNA polymerase, the enzyme we learned about just a second ago, DNA polymerase moves down to 3 to 5 on its own. Just by virtue of its shape, it has no issues moving down a 3 to 5 strand. So that strand, the leading strand, experiences continuous synthesis of new DNA. So it starts at one end and finishes. There's no hiccups, there's no fragments, there's no nothing. All right, it starts at one end and finishes, and at the end you have an identical copy of that original DNA molecule. Back to the replication fork. The other strand that goes 5 to 3, again, it's anti-parallel in nature. The other strand that goes 5 to 3, sorry about that, that has to actually work still coding in a 3 to 5 direction. How can we do this? Well, think about driving down a one-way street. You can only go one direction. That direction, we'll say, is from 3rd Street to 5th Street, from 3 to 5. So what if you miss your turn? Well, you have to hit a block, so to speak, or turn somewhere, go around, go back down, and get back on the street where you first started. So to do that, the DNA polymerase on the lagging strand will actually skip down the lagging strand. If it's already 5 to 3, as you can see right here, already 5 to 3, it will skip down towards the 3 end and encode back towards 5. And then skip down again towards the 3 and code back towards 5. And skip down again towards the 3 and code back towards 5. So it skips in giant loops or fragments or snippets, whatever you want to call it, but doing so creates fragments called Okazaki fragments. Okazaki was a husband and wife Japanese molecular biologists, or a team in Japan that ended up making this discovery and naming it after themselves, obviously. So these fragments, and remember, it has to be one identical copy of the original molecule. That's happened already pretty easily on the leading strand. The lagging strand, on the other hand, has now got these fragments. These Okazaki fragments are snippets or giant loops of DNA that have to be glued together. So that's where a third enzyme comes in. Again, ACE is an enzyme, and this is ligase. So DNA ligase is an enzyme that comes in and bonds different snippets or fragments or loops of DNA, chunks of DNA together with those phosphate groups. So you've already got your carbons bonded together, right? The sugars. Then you've got the bases out in the middle. So to get one nucleotide bonded to the next, you have to use this DNA ligase enzyme, and that puts a phosphate or a phosphodiester bond in its place. So up and down, the sugar phosphate backbone is joined to phosphates. In the middle, the rungs or stairs of this model or analogy, it would be joined with hydrogen bonds. So let's look at this diagram. Here's the leading strand. Notice it goes from 3 to 5. So it's able to continuously synthesize DNA. The green something to call a primer. Don't worry about that. But it latches on and it codes really easily all the way down. You can see in the next one, it's gotten farther because the replication fork is speeding ahead because helicase enzyme is just powering through breaking these hydrogen bonds. However, the lagging strand, named thusly because it's lagging behind, they skip ahead to this side towards the 3. They skip to the 3 end and code back towards 5. Here they skip to the 3 end again and code back towards 5. All the while, this one's going 3 to 5 continuously. Here you skip to the 3 end and code back towards 5. Skip to 3 and code back towards 5. Doing so creates these fragments called Okazaki fragments. So that last enzyme, DNA ligase, comes through and glues them together. Same thing here, but just another diagram, slightly more complex. So in class, we talked about slinkies. Imagine the entire escalator side by side. One goes up really easily. We say it goes from maybe the 3rd floor to the 5th floor. The other, remember, you can only ride one direction, but the other is going the opposite direction. So you have to skip ahead and then ride back, and then skip ahead and ride back. That would be the lagging strand. Same thing on a treadmill. We talked about these in class. In summary, you've got DNA replication. There's crash course videos, an explainer from the BBC. But again, the leading strand goes 3 to 5, so DNA polymerase is able to easily code down that. By code, I mean it matches complementary bases A to T, G to C. Whereas on the lagging strand, it goes anti-parallel. So it goes from your 5 to 3 carbon sugars in the opposite or anti-direction. So to code there, DNA polymerase still only goes in a 3 to 5 direction. It skips ahead to the 3 side and codes back towards 5. Skips farther ahead, codes back towards 5. In the end, you've got different fragments that are glued together with ligase. So the final product is still 2 identical copies of that original DNA molecule.